Okay, good morning, right? It's Tuesday, just to get everybody scheduled, because I thought it was Wednesday. At the beginning, let me read the official housekeeping rules. I'm sure you've heard these once or twice. Please silence your phone. Photography of ABRF meeting sessions is permitted, subject to speakers' requests not to capture pre-publication information. Are the speakers okay? Perfect. Recording of the program sessions will be available on the meeting website following the annual meeting. Social media policy is that everything can be shared. If a speaker asks not to be shared, please respect the speaker and do not post any on social media. Hashtag for the event is hashtag ABRF 2025. So if you're on Blue Sky, please be posting. Unless noted by the speaker, questions are to be held at the end of the session. Attendees may ask questions at the microphone in the room. If you haven't yet, download the ABRF meeting app to view the full schedule, access Lightning Talk abstracts, connect with other attendees and sponsors, play the ABRF game, and create your personalized meeting schedule. You'll find extra content in hours of the exhibit hall including hours of the exhibit hall and tech showcase to thank our sponsors. All attendees will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. Done. Hello. My name is Tim Bushnell. I'm the director of the Center for Advanced Research Technologies at the University of Rochester Medical Center. That is in upstate New York. We are really the home of buffalo wings, even though Buffalo claims them. So don't let that fool you. I am pleased today to be hosting this session called Pathways to Single Cell Analysis. The ultimate goal is this is to be a discussion, interaction. Our three presenters, Steve Polter, from the University of Rochester, Jeff Malik from the University of Rochester, sense a theme here, and John Preall, who I will recruit to the University of Rochester at some point, but he's currently at Cold Spring Harbor.
My brothers live in Rochester.
Hey, there you go. That's close enough. They represent flow cytometry, genomics, and bioinformatics, and the whole idea is how do you take a cell in a tissue and get actionable information at the end? And there are steps along the way, and that's what we want to talk about. I will mention that Dave Haviland was supposed to be here as our flow cytometry speaker, but unfortunately he had a family emergency, and I picked on Steve, and he graciously volunteered. So the speakers will get a few minutes to introduce themselves, tell you a little bit about their philosophy and theory, and we'll then open it up to questions and discussion. Now, to begin, this session is sponsored by Parse. Are you Parse or Parse Bioscience? Parse Bioscience, thank you. I just wrote Parse in my notebook. And John Thompson, that's an easy name for me to say, who is not at the University of Rochester, is going to talk to you a little bit about their technique, smash the limits of single-cell sequencing. John, why don't you come take it away?
Thank you, Tim. Unfortunately, I'm going to have to break your heart a little bit. I like my Bay Area a little bit too much to move to Rochester. With that out of the way, hello everyone, I'm John Thompson. I'm the Parse Field Application Scientist from San Francisco, based in San Francisco. And I help Parse users run their Parse kits and help them smash the limits of single-cell sequencing. And so, let me make sure this is working. This does not seem to be working.
Does that have to be tried? Let's see.
It's on.
It's on?
It is on. Apologies for the technical difficulties. We'll figure this out.
Our technical person left. Oops. Oh.
All right. Lisa, can you hit next when I'm ready for the next slide, please? We'll do this the old-fashioned way.
I thought they were going to have a computer up here.
I thought so too as well. There we go. Okay, I'll just say next when we need the next slide. So some of the greatest strengths of parsed EverCode single-cell RNA-seq technology is that it is scalable. So you're using the same workflow for our smallest size kit of 10,000 cells total as our largest kit of 5 million cells total. And these are off-the-shelf kits that you can run in your lab either by hand or using automation options, but you don't need to purchase any capital investment, no instrument required, there's no service contracts. All you need to do is be able to pipette, use a multi-channel, use a thermocycler, use a centrifuge, standard things that you'll have in your lab already. Unmatched data quality, we see higher gene and transcript sensitivity. You avoid doublets, you avoid ambient RNA, and the biggest strength is that it's powered by fixation. So that locks in the biology, and it also allows the researcher to prepare the samples in collaboration with the parsed field application scientist to make sure that they have good quality samples. They can fix the samples, freeze them down in the -80, cryopreserve them, make multiple aliquots, send them to you, and then you can do additional QC. And you can fix samples, and they're good for up to 6 months. So, first you'll fix the samples on your schedule, or the researcher's schedule. Then, once they are done with the fixation and all the samples are ready, they can easily deliver the samples to your lab, either by walking them down the hall or shipping them across the country on dry ice. No frantic running into the lab at 9 p.m., here's this fresh sample. No, they'll deal with the fixation themselves, and then they'll deliver them in a timely manner. Then, likely the core will do the barcoding and library preparation, which allows you to attach a barcode to each cell, and I'll go into that in detail in a little bit. As far as sequencing goes, Parse is compatible with all major sequencing platforms. Natively, we're compatible with Illumina short-read sequencing, but also there can be conversions to Ultima, Element, Singular, MGI, etc. for other short-read sequencing platforms, and of course, long-read sequencing on PacBio or ONT platforms. And then, for the data analysis, you can either work with a bioinformatician, or you can use our user-friendly Trailmaker data analysis that allows you to demultiplex, align your transcripts, and then do the data analysis from FASTQs all the way to publishable figures. And for the bioinformaticians in the room, this allows the biologist to do the analysis, do the simple things, and frees you up to do the complicated and more fun analyses. This also allows you to share data super easy with the researcher, collaborators, anybody who needs to take a look at the data. So, the way Parse technology works is we use the cell itself as the reaction vessel. That means that we have a fresh single cell or single nuclei suspension, and we will fix the samples, permeabilize them, and then cryopreserve them in the -80, and they're good for up to 6 months. So this allows us to lock in the biology and, importantly, transport the samples safely from the researcher's lab into the core lab. And so the way that EverCode combinatorial barcoding works is that you'll have your fixed and permeabilized cells. You'll load them into the wells of this first barcoding plate. So that's our first split. And each of these wells has a well-specific barcode number 1. We'll incorporate this barcode by reverse transcription, appending this barcode and turning your mRNA into cDNA. And since we know which well has which barcode, we know which samples we loaded into the well. Therefore, this is a sample-specific barcode. The next step is we'll pool all of the cells together from all of the wells, and that inherently multiplexes everything together. So there's no cell hashing, there's no inefficient multiplexing. The multiplexing is built into the technology itself, and that allows us to multiplex 96, 384, etc. samples in one experiment. Then we'll do a split, randomly distributing our cells into the second barcoding plate, ligate on our second barcode, do that again, pool, split, ligate on our third barcode, pool, wash, and count our cells, which allows us another QC step before we go into our sublibraries, and then split them into what we call sublibraries. For here, this is a mega kit. And during the library prep later on, we'll add a sublibrary specific UDI barcode. So in this way, barcode 1 is a sample specific barcode. 2 and 3 add complexity and diversity. And 4 is a sublibrary specific barcode. And this is one of the biggest strengths of parse also, is these sublibraries. So with other single cell experiments, you throw cells at it and you hope you have the right number. With parse, you're going to normalize the amount of cells here when you count. You're going to also QC and then load your sublibraries. And if you're loading a certain amount of cells, that's how many cells you're going to get data for. So if you load, let's say, 12.5 thousand cells in here, you know exactly how many reads you want to throw at it to get 20,000 reads per cell. Less guesswork, and you can also sequence one of these sublibraries first, look at the QC, and since everything's multiplexed together, you'll see all your samples. And then later, you can go in and sequence the optimal number of reads for the rest of your sublibraries. So, with this, we have over 14 million barcodes for our mega kit, for example, our 1 million cell kit, which allows us significantly lower doublet rates, usually about 1.5 to 3%, regardless of kit size, whether that's 5 million, 1 million, 100,000, et cetera. So you can fix the samples. They're stable for up to 6 months. And what that really enables, and like I said previously, parse can send the fixation kit to your researcher and send the core, the barcoding and library prep kit. When the researcher is done, the researcher will get comprehensive training on how to fix the samples, how to QC the samples with a parse field application scientist. If you're in the Bay Area, that would be me. And once they learn all the QC, once an FAS has looked at their images and said it's all okay, then they will send the samples to the core to run it at your leisure. Now, another option which is great for cores, and I see a few happy faces in the crowd who have automation options for parse. So parse is automation friendly, and what's great about parse automation is we're platform agnostic, though we do have some preferred collaborations on automation. So what's great about automation is it's predictable. With so many samples, human error happens. So with automation, you get minimum variability, more reproducible results, more predictable turnaround time, and of course, logs that can increase the traceability. Scalable, with working with more samples, it's a little bit more difficult, and this allows you to easily scale up to more samples without having to increase the stress on your core. And of course, it's very cost effective, increases your reliability, less repeats, and optimizes the process. So we have three different options here. We have a plug-and-play Integra Assist Plus, where Parse works with Integra to get you an Assist Plus. We'll set it up between the Parse FAS and the Integra rep. We'll go right to your lab and teach you how to use it. We also have HTS partners, which can help you set up these other platforms. And so if you're interested in barcoding the workflow with the Integra Assist Plus, from fixation, barcoding, and library preparation, stop by the Parse booth or the Integra booth on the vendor show hall. And in conclusion, Parse has unmatched data quality, better gene and transcript detection. You avoid doublets and ambient RNA. Flexibility is a huge thing. The researchers fix the samples, send them to the core, and you run it on your time, not frantically at the last minute. And of course, it's exponentially scalable. You can run 1 million, 5 million, 100,000, whatever fits the research. And these are the different kit sizes. We also have additional profiling methods as well. If you want to learn more, please stop by our booth at 108 on the vendor room hall. And I'll go ahead and turn it on to the next speaker.
Great, thank you, John.
While the presentation.
Next, we'll have Steve Polter talk a little bit about flow cytometry.
Thank you, Tim.
Once we have our slides.
Yes.
There we go.
Wonderful. Hello, hi, I'm Steve, as you all may know by now. And I'm here to talk about the spot that flow occupies in the pathway to single cell, which is often the sort of first step in getting your cells to that process, right? So we go hand in hand, right? You use flow to explore your samples, your populations, identify populations of interest, drill down, and finally sort out the cells of interest, send them to single cell. But there's a bit more than that. Perhaps deceptively simple based on the description. So we're natural partners, right? But with that natural partnership comes natural challenges. So I've broken it down into the three main challenges that we see, laser, and some of the sub-challenges within those, right? So you want sample cleanliness, purity of sorted populations of interest, and viability of sorted cells, right? All of these things come together to produce a sample that is either workable for single cell or not workable, as I am sadly familiar with on both sides of that. So with sample cleanliness, right, optimizing your cell prep is the first thing you want to be thinking about. And this is all stuff you should be thinking about before you start putting cells into tubes or, getting things together and sending it to the genomics core, right? Because I've encountered a lot of scenarios where people are like, yeah, we're just going to throw this into the single cell, pathway. And it turns out that your sample kind of sucks, unfortunately. Not to put too blunt point on it. But you want to minimize your debris, right? And that can be tough if you're doing a tissue digestion. You want to dial that in. You basically want to find the minimum point at which you need to be to get your cells separated from that tissue. You want to also minimize cell death, right, which can be tough if you're digesting, if you're working with a sensitive cell type, sensitive tissue type. So this is something that's a bit painful, right, because it can take a lot of time to optimize for this. Spinning as gently as possible without losing excess amounts of cells. A lot of these things that I'm going to be talking about, you have to strike a balance, right? And that's going to be different for every project. It's going to be different potentially for every investigator, for every cell type. So it can kind of be a pain in the butt. And a lot of people don't necessarily want to put the legwork in at the forefront to make sure that you have quality product going into single cell sequencing and coming out of single cell sequencing. Right, so as an example here, enrichment, not as an example, but enrichment is something that I always recommend to people. It's something that some folks do at the outset, something that some folks never think about. But there's a lot of ways to enrich or deplete your samples to kind of get rid of the dross, right? The things that you don't care about. Separate the wheat from the chaff. You can use magnetic sorting or columns. Density gradient, right, immunoaffinity, so on and so forth, right? So there's a lot of tools available in our toolkit, and I'm not going to enumerate all of those here because that kind of strays away from the point. But there's a lot of tools in our toolkit that allow us to get a higher quality input, and it's something, again, you have to put the legwork in to explore. So it's probably a point I'm going to belabor, and I beg your pardon ahead of time for repeating myself. Your purity of sorted population, right? So your fluorescent panel is paramount. If you are sorting cells on a cell sorter, you have to be able to identify the population that you want to sort reliably, right? And that, again, is something that can take some work to figure out and optimize. From the standpoint of what markers am I going to use to, oh no, my fluorochrome and marker pairing really doesn't work very well for this super rare population that I'm looking for. So using controls, right, putting the work into design, striking a balance, again, between complexity and ability to resolve the population of interest, right? Because you want to have the minimum amount of colors you need for all sorts of reasons, right? It's less work, there's less money spent on reagents, but you want to be able to get that thing that you're looking for. And again, optimizing panel to maximize ability to identify rare populations. I can't stress that enough because, if you have a poor pairing of fluorochrome 2 cell marker, it's just not going to work out well. Setting your gates to exclude your debris, right? Some people go, oh yeah, I can just gate out all the debris, right? No, that stuff overlaps hardcore, right? If you've ever run flow cytometry, there's a lot of things that overlap, doublets and singlets, the cells that you want and the cells that you don't want, and the stuff that isn't cells that you don't want. So you can gate stuff out. Don't make that your only point at which you're going to be getting rid of those things. Or the debris really is what can be problematic there. And we see a good example of that in my quickly pulled example gating scheme. But at least the populations look halfway decent, right? You want to be exacting. I get a lot of investigators who go, we're gonna widen that live dead gate because I want enough cells to go into my tubes, right? Well, then you're sorting a bunch of crap that isn't worth your while, right? And when the GRC folks go to QC it, they pop it on a cell counter and they go, yeah, 70% of this is debris and non-cellular junk. You don't want to be in that spot, right? Because that's disappointing for everybody, mostly you as the investigator, I would hope. Now, one thing I want to backpedal very quickly here. Sample cleanliness, one thing I don't mention on the slide is use a cell counter before you get to the GRC. They will do QC. They will make sure they're not wasting their time, your time, anybody's time. But if you have access to a cell counter and you can verify the cleanliness or the quality of your sample or just see if it's not even good enough to bother going forward, that's excellent, right? Because that can then take you back to square one, unfortunately, right? You got to go back to the lab and you have to optimize. You have to figure out what you're going to do next. Viability of sorted cells. Sorting is inherently a stressful process. There are some exceptions to that, which we will talk about. But if we're talking about an electrostatic cell sorter, we're working at potentially high pressures. We're zapping those poor little cells. We are hitting them with lasers. Not all in that order, but it's pressure laser zapping, right? And then you have decompression as well, which is something I hadn't thought about for a while until, Peter Lopez presented his talk on sorting-induced cellular stress. So it's a big thing, that's a big consideration. Now, the larger pore size we use in nozzle, right, the lower pressure we're working at, so we can get away with reducing that stress. It depends on the cell type you're working with. You know, mostly the sensitivity, right? Also, how large your cells are, right? You don't want to clog the sorter. So I usually recommend a larger cell or pore size for anybody who's doing any kind of work that has any amount of sensitivity from cell culture to single cell sequencing, right? You can pad your odds a little bit by increasing the volume of media in your collection tubes, using larger collection tubes. Now again, this is something where you have to strike a balance because you don't want 3 cells and 3 mils of media. If you spin that down and try to extract them, it's probably not going to go well for you. I haven't tried it, so don't quote me. But now, microfluidic cell sorters, right, are kind of an exception to this. So they run at a lower pressure, but they also have some limitations, right? Some of them don't have as many lasers, as many detectors. If you're sorting multiple populations, you may be limited by how many sorting valves or paths you have, right? As we can see in this example, you've got 2 sorting pathways. If you're trying to do more than 2 populations, you're out of luck, right? So you could either do that as an iterative process multiple times or make some concessions, which we all have to make at some point or another. Let's see, is that on the next slide? No. Now I do mention here, and sorry for going back and forth, I mentioned here pre-sorting with a microfluidics device. That may not always be viable or desirable, right? I popped that up there because it is a consideration depending on the instruments available to you, the techniques, the people, etc., right? But there are a lot of ways to get your purity up, right, before you get to that sorting process. I just wanted to take a moment to talk about that. What else can we do to improve, right? I've talked about some of the technical stuff, some of the things you can do hands-on, but what else can we do? And I think the biggest thing is coordinate with your genomics folks, right? Jeff and I often talk when I have an investigator, or he has an investigator who wants to do single cell. Sometimes they come to him first, sometimes they come to me. It all depends. But the biggest thing that we've started doing is giving each other a heads up, right? I look at an investigator that says, I want to do single cell sequencing at the end of this sort. And I go, okay, when you coordinate with the genomics folks, you're going to have me and the rest of my team on that email because the genomics folks and I want to know what's going on at every step of this. And if any one of us has suggestions, questions, etc., the more people that are involved in this process, they're in that communication, the better we can potentially do, right? Because we don't have mix-ups, misunderstandings, snafus. I mean, those are always going to happen, but you want to try to minimize that, right? Having everybody at the table, right? So the concept of the round table. At this particular round table, there is no king. There is the investigator who ideally has the wherewithal to get everyone together. Sometimes that has to come from the flow folks or the genomics folks, though, depending on who we're working with, what their experience is, etc. And I certainly don't want to sound like I'm talking down to anybody there because we takes all types, you know, at all points in their career. And we're here to help, right, as part of being in a core facility. I think that's probably one of our prime directives is to help without judgment. Maintain that channel of communication. Instant messaging, we have Slack. We have a GRC flow core Slack where we just slap messages back and forth pretty frequently, you know, we will share plots hot off the sorter. As we just finish a sample, we will send that sort report, we will send those plots, we'll say, GRC folks, here's our plots, here's the sample, what do we think? Do we have any questions? Do we want to reinform? You know, we might have more sample, we could potentially resort, change some gating strategies. What's up? Instant messaging, real-time updates, and reducing downtime right between the end of sorting and starting the single cell analysis, which you don't want to waste valuable reagents, et cetera. You want to be prepped. And with that, I will conclude. Thank you very much for listening to me.
Okay, our next speaker is Jeff Malik, also from the University of Rochester. He represents our genomics team.
Hi everyone, hope you're enjoying yourself here. Thank you, Tim, for inviting me to do this. And thanks, Steve, for pinch hitting.
Is that yours? No.
Nope.
Malik?
Nope.
We should have a meeting where we have random slides and everyone, you have to speak on a...
Oh, that would be so fun.
Just ad lib.
Done that on the grad student group.
There we go. Thank you.
Hilarious. Absolutely.
So as Tim said, I'm Jeff Malik. I'm the technical director of the Genomics Research Center at the University of Rochester. So I'll talk about how we fit into the single cell workflow, handing off from, after the handoff from Steve. So just a little about me and us. So I'm a New Yorker from birth, Long Island originally, made my way up to Western New York through Geneseo as an undergrad, and I spent some time after that as, let's see, figure out the laser thing here. Thank you. Anyway, so then I spent a few years as a lab tech at the U of R, trying to figure out, did I like this lab life? What did I want to do? And it actually set me up well for where I ended up. Did a lot of molecular biology. I ran some sequencing gels. I meant to bring an autorad here, but I couldn't go back to my old lab in time to grab one. And ultimately that led me to grad school. I got my PhD, and there is when I kind of started to interact with Tim. So he was setting up the flow core, which would ultimately turn into CART. And I did fair bit of flow cytometry. We did some microarray data analysis, and then ultimately went off, did my postdoc at the lab next door, which got me into high throughput sequencing techniques, mainly ChIP-seq, did some RNA-seq. It kind of set me up to kind of where I'd kind of jump off, and one day found myself talking with John, our core director, saying, hey, have you heard of any jobs around? He said, actually, we're hiring. So that was about 8 years ago. And now I'm at the Genomics Research Center. Like I said, we're part of CART, so the Center for Advanced Research Technologies, with Tim heads up. We're also a member of our Wilmot Cancer Center shared resource, and we just got NCI designation officially this week. But just to say, we're well integrated with the other cords. We're constantly working to reduce the friction of that handoff for investigators who may be working with one or more of us, which is very important when it comes to single cell analyses. So within the GRC, we have a unique setup. We're kind of like a three-headed monster at the top, with John being at the head and the head monster. Sorry, John. Elizabeth and myself, we kind of run the day-to-day operation. I'm more on the hands-on, bringing on new technologies, troubleshooting, working with the staff to help iron out issues as they come up. Elizabeth's more big picture, making sure the trains are on time, and constantly communicating and scheduling with investigators. We have 8 or so staff. This is a little outdated on the wet lab side. We have about 10, 8, 9, 10 bioinformaticians as well. So we kind of do everything from sample QC through analysis. So we have a pretty comprehensive, standard report we send after sequencing. Kind of a not meant to be exhaustive list of our services, but everything again from bulk assays, RNA-seq, whole genome, epigenetic type stuff, single cell obviously, which we'll talk about today, and spatial. And here's some examples of the things we have. And again, talking about single cell specifically, so we are heavily invested in the 10x platform. We also do things like SmartSeq on sorted cells into plates. Along the lines of the 10x, we are using hashtags and CiteSeq constantly with investigators and whether when it's suitable for their needs. We also have done single cell ATAC and multiome. Today I'll focus mainly on the RNA and CiteSeq hashing flavors of that. We're currently testing Illumina, Parse, other single cell methodologies to kind of just broaden our services and be receptive to the community. Because that is important, right? And it's not a one size fits all. There are gonna be specific assays that are better for one cell type or one investigator compared to the other. We've evaluated, Hive, BD Rhapsody, earlier versions of the Parse and Fluent technologies as well. So we're definitely open to that. We're actively investigating those things, but sometimes it just doesn't work out for one reason or another. Okay, so why single cell? I don't have to tell everyone in this group, but it really is to drill down on and measure the gene expression, in this case for genomics and transcriptomics, the most basic fundamental unit of life. And that's for cell typing, to define cell states, do differential expression amongst those groups. You can also branch out into epigenetics, like I talked about, single cell ATAC. We've done some testing with investigators who want to use single cell cut and tag, leveraging the capabilities of the 10x pipeline. And then other features which are related to flow, such as cell surface antigen expression through CiteSeq, or for immunologists for looking at immune specificity for the decode dextramers. Kind of, I'll be highlighting an example of an experiment. We had a lot of touch points with flow and MUS, which we use these methodologies. And as Steve very well described, sample prep considerations. It's the first thing we talk about with investigators. It's usually a very large discussion. It can be a lot of back and forth, a lot of optimization, and sometimes investigators aren't always receptive to those, to that amount of work that may be involved, that level of optimization. But we try our best to work hand in hand with the investigator, with the flow core, to help get to the best sample possible before we move into our single cell assays. And that can, like Steve said, tissue dissociation, using flow sorting to remove those debris, the dead cells, et cetera. And then in general, things we look at once we actually get these samples, like Steve said, we get the cells on a slide. We do trypan count, look at live dead, maybe use some fluorescent, use our fluorescent counter, and at times manual counting is needed. If you have a particularly dirty cell population, like I'm showing on the upper right there, that's a bad sample. We would not want to capture that with any single cell technology. You're gonna have a lot of debris, potentially clog it if you're using the 10x system. And on the bottom is one that's been well processed. So we have single cells, very few dead cells, no clumps, no debris, et cetera. And there was a very good talk yesterday that kind of explained the whole ambient RNA effect. If you have lyse cells, RNA goes into the solution. It's gonna go into your droplet. It's gonna go into your other microwells if it's there. And it just causes a background issue at the end. All right, so I'm gonna kind of get into, lay the groundwork for a good experiment. I thought it would be nice to highlight the sort of how we think optimally things should work from the investigator through a flow sorter, through the flow sorting facility to us. And this, CiteSeq and hashtags, again, looking at the cell surface repertoire at the protein level. Thank you to friends at New York Genome Center for about 8 years ago publishing the paper on CiteSeq and showing that we could oligo tag antibodies a la flow sorting. Instead of having fluorophores, we now have oligo tags that can be read out on genomics platform, but can very nicely measure, like these histograms showing what looks like to those who do flow plots, right? So it's sort of a very familiar way to output the data, but leveraging again the sensitivity and the abilities of the sequencing technology along with the transcriptomics. And hashtagging allow you to label cells when you have small numbers of cells to be able to cobble samples together so you have enough sort of critical mass so they move forward with your single cell experiment. So using hashtags, again, the oligo hashtag antibodies, so we have different molecular barcodes read out by the sequence of the ATCs and Gs. predefined by the investigator. You label your cells before you go to the flow sorter, and then those hashtag antibody labels survive the sorting process. They then can be pooled later to go through and be deconvoluted after analysis. And this can be done to reduce costs, to help define doublets, things like that. Again, when you have low cell numbers, it can be very difficult for us at the GRC, at the Genomics Center, to quantify that sample, measure the cell concentration, and then accurately get this number of cells required for the downstream analysis. So many times we're using event counts, again, with our communication, with the flow core, receiving those event counts, being able to pool samples based on those those event counts, to then be able to predict the number of cells we're likely to get out at the end. And we have to make some assumptions, because not every event is a cell, and we kind of work with my experience with the flow core over the years, we kind of settled on this 80% sort efficiency. So for every 10 events, we assume there's about 8 cells. So here's an experiment that I thought was nice to highlight. So this investigator, he's one of our, I would say he's a pleasure to work with. He takes all our advice. He works well with the flow core, does our sort of testing experiments to make sure that the flow panel will work, to make sure you're getting good quality. This was a very complicated experiment. So he's looking at sorted human T-cell subsets from premature babies at term and then adult samples. So these are very precious samples. They're coming out of the freezer. 4 populations sorted per subject with 4 subjects per experiment. So we have 16 populations. And then he had a very specific pooling ratio he wanted to target for each of those 4 populations within each subject. And then he wanted equal representation of each subject. So 10,000 cells, let's just round numbers, 10,000 cells per subject, but within those 10,000 cells, the 4-to-4 to 1-to-1 pooling ratio. Ultimately, he wanted 30,000 cells captured per target. For this particular experiment, we're doing 5' GemX from 10x. So this 30,000, went the wrong way. 30,000 cells is actually a super loading of the GemX platform, but something we wanted to test for him and get his, get nonetheless. He's also using hashtags, doing CytSeq for about 25 different antibodies, and using decode dextramers to look at immune specificity. And this is sort of part of the readout we get from 10x report, so I just kind of wanted to show you what we're looking at, the complexity here, because every time he brings us samples, we're getting 16 tubes, and we now have to make this work. And luckily, when a plan comes together, when the investigator is receptive to all of our notes and suggestions, we have good communication with the flow core, we have good event counts, which are kind of the second line. So this is my chicken scratch, because I was heavily involved in getting this experiment working. But at the end of the day, we were mostly able to get this to work. We couldn't quite get 30,000 cells for all of his samples, but we nearly nailed the ratio that was important to this investigator. And that says a lot about how Flow is able to get the events that they required, our workflows over the years to make sure we got our cell concentration down, because we're dealing with larger volumes off the sorter, et cetera. So a lot happened. Here's an example of some of the CytSeq antibodies and their profiles and that we were able to get after sorting as well. So success story when we do well by talking within the cores. Oops, sorry. So coordination between shared resource labs is essential. I think luckily, since Tim asked Steve to fill in here, we were able to highlight something very specific between the U of R cores. But ultimately, again, like Steve highlighted, this is about reducing friction for the users, right? We're here to help them, and we never lose sight of that. And we're ensuring sample quality, right? These are sometimes rare samples, very precious samples, and we want to make sure we have a chain of custody that's keeping that quality high. And importantly, we're leveraging the expertise of each individual core. I'm lucky I have a pretty good background in Flow as a user in my graduate student years, so I can speak the language enough to make sure details aren't missed. And we have several folks in the GRC staff that also have that experience. So I think some of that may be unique, but nevertheless, I always point towards the flow core expertise when needed and never overstep my comfort level. And this is important, beyond flow and single cell transcriptomics, multimodal data collection integration. I can't speak to the integration between flow data and transcriptomic data, but that's coming, right? I think folks are gonna need, in this day and age, with limited dollars, be able to take a single sample, measure it multiple ways, and kind of look at the fuller picture. And how we do that, I think, at least in my mind, is a bit of an open question, and then there's gonna be multiple ways to do that, I'm sure. And then also just, 1 + 1 = 3, right? This is added value. We're using what we're good at the GRC, what Flow's good at. We have proteomics represented in the audience, right? Each of us have our things that we're experts in. Let's bring that to the table and help the end user for added value. So with that, thank you for everyone for listening, and I'll sit down.
So our last introduction is John Preall. Am I saying that right?
Preall? Yeah, I mean, I'm used to it just being a lot of Preall.
Okay. From Cold Spring Harbor, who is our informatics expert, here to tell us how we actually deal with this data.
Okay, so my presentation is going to look suspiciously like I took a lazy way out and didn't make any slides. But I will assure you this is for good reason. I've given a couple of these before ABRF and recently in Albany. And I made a nice 15-minute presentation. And it turned out, at the end, everyone walked away. And I felt like nobody took anything from it. So I want to encourage strongly audience participation here because we need to leave time for this. So, okay, I'll talk a little bit about bioinformatics, but I think I want to make sure we leave some time for discussion. All right, so my core is actually, I'm at Cold Spring Harbor, and my core is comprised of 3 people. And 2 of us have been here the entire time since we started in 2017, and we are a soup to nuts core. Actually, yesterday, Walter Eckelbar introduced himself as a co-lab at UCSF, and I really want to steal this concept. We are not really a core. We are a collaborating laboratory, and we do everything from the flow cytometry. We plan your projects, we do your flow cytometry, we do the genomics, we do the bioinformatics at the end. And Cold Spring Harbor has the luxury of doing this because there's only like 80 labs or something like that at the whole thing, and we only work with a few of them at a time. So we don't have the kind of throughput that you guys have to deal with that just like turn projects out all day long. So I'm involved with almost every aspect of a project. And actually, I just second every single thing that you guys said. Absolutely everything is correct about sample prep and whatnot. But in terms of bioinformatics, I think the pathway to single cell analysis has to be a unique solution that's really dependent on each individual institution and how they approach bioinformatics. And from what I gathered from talking with a lot of the people here at ABRF and my other colleagues elsewhere, bioinformatics is in an odd state right now where there's massive brain drain. And a lot of the people that we would want to hire to be staff bioinformaticians are leaving for better paying jobs in industry, and I don't blame them. So at Cold Spring Harbor, in fact, we've had a bioinformatics shared resource that has had a dubious history over the last 10 years or so. It's shrunk, and now it actually disappeared for a few years, and now it's back in the form of having 2 employees, one of which used to work for me, actually. And, but now he's an independent director of the bioinformatics shared resource. But they don't, obviously, 2 people don't have the capacity to do all the projects here. And so instead, we pivoted to a different angle that I have heard other institutions, small and large, doing as well, where we're taking an education-based approach to doing bioinformatics. So we can hold seminars, we can hold workshops, and we can build tools that are accessible, and we train postdocs and students to come in and use them and interact with their own data. So in my history of working with people's projects, I've done sort of all forms, right? I've done projects where I am the point person on the bioinformatics and I'm simply doing all of the coding. And that's fine, and we've produced papers this way. But by far, my preferred way to do it is to be a mentor and a collaborator to a student or a postdoc who's doing some of their own work themselves. And it's our job as bioinformaticians at Cold Spring Harbor, we're building tools to reach out and make that a little more accessible. So I don't have slides for this, but trust me, they'd be boring anyway. So Cold Spring Harbor has built, Rad, under the director of the bioinformatics shared resource that I mentioned a second ago, has built a set of Jupyter notebooks for workflows. Single cell, your standard, ScanPy tutorial plus a couple of extra gene set enrichment stuff that everyone does all the time. A couple of these standard workflows that you can just plug your data into, and then it's linked up to the way we store data on our cluster and whatnot, so people can access it and run those things. In addition to all those weird bulk things that some people do that's not single cell. So he's built a lot of this, and I think he's learned the hard way that building and maintaining a large code base like this is really, really challenging. But nonetheless, it's a useful tool. And we have workshops for Python and R and things routinely to do this. I recognize that there's a lot of other strategies that institutions could use to bridge the gap. Like for instance, there's commercial software, Partech, et cetera. Illumina is going to be, I'm not sure what the cost structure on this, though Illumina's implementation of Partech is going to be. But Cold Spring Harbor has made the choice over year after year to not invest in any of these platforms because they're quite expensive, and ultimately it doesn't really produce a much better result than learning how to do it yourself. Okay, so the single cell analysis has all kinds of challenges and things that we've had to teach people to work through, but the most important hurdle we think is actually getting people to formulate a question in English language that they're going to ask of their data. And what we have found as well is that if you can do that, then your pathway to analysis will be actually cleared for you, right? Because you can work with a bioinformatician, or you can work with AI models now to write your own code, and ultimately get there. But the bottom line is, if you approach your single cell experiment with a mentality that you're going to see what you see, then I will not work with you. I'm not interested in this kind of project. These days are over. There was low-hanging fruit for 5 or 6 years where we could just publish papers like this, but I think the field has moved beyond this, and you need to approach your single-cell data now. If you're going to publish in anywhere worthwhile, you need to have a better approach. And if you can't state it in English, then you can't state it in code, and then you're not going to get anywhere. And so I've learned to avoid these things. And thankfully now we have language models. You can just state your problem in English and get a lot of the way through it. And so that's another thing that I've actually found has been extremely useful, is that we work with students who are extensively using, all of the various language models, to help facilitate code, write boilerplate. And then it's critical in these situations for these students who are, uninitiated in code to come check with people who have a skill base and actually an education on this to make sure that they're not making any egregious errors. But for the most part, having relatively low expectations of what AI language models can do for you, and treating them as a way to essentially, skip all of the Stack Overflow reading that you're going to be doing and get some of those calls done has been an enormous boon to bioinformaticians everywhere. So that's kind of really where I wanted to start the conversation. There's not much else I could say here. So I'm really curious whether people can have any insights on how their institutes are solving the problem of the lack of bioinformaticians that are available to them. But otherwise, I think there's probably questions opened up to, for all the topics here. So we should take time to do that. All right, thanks.
So this is a fun audience participation part. So we have about half an hour before lunch, so you got some time to kill. Mike, if you have questions about any topic, flow, genomics, or informatics, or the combination, there's a microphone there, or you can shout if you're loud enough. And I'm going to tell you, if I know your name or I know who you are, and you're not answering, I'm going to point out to you and I'm going to call you out. So I've got my eyes on you.
So that was great. Is this on? Does it matter? Okay. So I have a question, and I think you'll know who I'm asking. In a bioinformatics service offering, I think that a lot of PIs and a lot of institutions are used to paying money for that analysis.
Oh, are they?
For
I
that
haven't
expertise.
experienced that.
Okay, yeah. It's fair point. It's fair point. In my institution, there's a lot more friction around education and paying money for that training and that different kind of support. I'm just wondering if that's something that you have a perspective on.
Everywhere is different, obviously, but first off, we have had the hardest time getting people to pay money for bioinformatics, and especially the price tag involved, right? You know, human hours add up really fast, and it takes a lot of human hours to do stuff, and sometimes they're not really the most glamorous stuff, right? You'd like to think you're spending all those 6 hours just like, producing these glorious plots, but it's like you're data wrangling and munging for the first 4 hours of it half the time anyway, and they don't want to pay for that. So that's been brutal, so that's one of the reasons why we shifted over to this other model. But Cold Harbor has a cancer center. We're a basic cancer center, and a lot of the cancer center initiative is meant to, people's salary gets partially paid by the cancer center, and that's how we've decided to justify it to the National Cancer Institute that certain percentage of my time and all the other people's time is going to leading these workshops and stuff like that. And so as a result, we don't have to charge individual PIs for this. We can just say, please come. And in the end, it kind of benefits everyone to, be a little bit self-empowered. And so I think it pays for itself. But, the answer is go get money from somewhere else. The federal government is a good place to go right now, I think. Yeah.
Hi, I'm Jane Shrivastava from the Gladstone Institutes. We are luckily, one of our institutes is data biosciences, and we heavily invest in a bioinformatics course, so we feel quite lucky for that. Just a question, especially in terms of the flow and the genomic side, and this is quite a simple question with a caveat that everybody's experiment is different. But when somebody comes to you and says they want to do single cell work, do you provide them with standardized SOPs, any kind of documentation or resources or anything like that? Thank you.
Yeah, good question. So yeah, it depends. I think we do have standard SOPs for things depending on if they need help tissue dissociation wise or getting good cell suspension. So yeah, we can definitely provide those. We will be as involved as they'll let us be, right? So we will give them we don't do a lot of these sort of max purifications or columns. We know which ones work well, so we'll kind of point them in those directions, spoon feed them as, whether it's a grad student or a postdoc that's working on this project. And we'll try to kind of help them along the way. We offer evaluation of the samples, up front ahead of time, so we're comfortable that the sample quality is going to be there when we need it to be. And in this way, everyone sleeps better, because once you have that precious sample and it's there and it's low quality, that's the best it's going to be, because the clock is ticking, and if they have to go now rush over, try to get a flow time slot to sort, that's going to add hours. We may be already close to the end of the day, and the cells may not survive. So we try to work very hard and prepare. We do get the situation where it's my mice are dying and I need to sort tomorrow, and can you help us? And we'll do our best to kind of clear the schedule or make sure people are available, get them on the sorting schedule. So we do have those situations. We like to be involved ahead of time, if not, hopefully weeks ahead of time, and do those testing evaluations when we can.
Yeah, to provide an answer from my perspective, I think this is actually one of our weak points at the Flow Core, because we don't have, I don't have a packet to hand to somebody when they say, I want to do single cell. Now, on the flip side, the people who are usually going to genomics when they're sorting are either already steeped in flow and they're going to make the mistakes they're going to make, and we'll address that when that happens, or they know how to do things like Adam, I believe, is whose work you were mentioning there. Excellent guy to work with. But I think, yeah, it's definitely beneficial to kind of have that info packet to drop on somebody, right? Because I do a lot of our sort consulting, you know, for people who haven't sorted before. So the SOP tends to be up here, and I just end up personalizing it based on who I'm talking to and what they want to do, which is great, but sometimes, you know, things may drop, right, depending on what day it is and, you know, what I've been doing the prior night. So I think ultimately, yeah, that's an excellent way to go, and I think for my own facility, that's something I should probably look into. But also, you know, as Jeff kind of mentioned, going from the individual route, right, and personalizing, I think that has its value too, because you're basically taking that and cradling it from birth to death, from start to finish.
And also on the industry side, reach out to the field application scientists for whichever technology you're working with. Miltenny has good service, or the single cell company you're working with will have good service to help you with that QC, those SOPs, those dissociation protocols. So leverage those resources that are built into the technologies you're working with.
Hi, I'm Dan Mielkars from Dartmouth. This is kind of for Steve, I think. So when someone comes and their sample is not great, do you involve yourself? Do you say you probably shouldn't proceed with this, or do you do your best to sort what you can? And then, you know, we've kind of leaned towards the last part, you know, the second option, and we sort what we can, and then we hear back from genomics like we didn't really get anything recoverable from this, or, you know, and then it feels bad from our end, and then it's kind of, they're blaming us, and, you know, for sorting, and but it's, you know, it's a sample that was clearly pretty deficient when we looked at it on the sorter. So I'm just kind of curious how you navigate. It's more of like an interpersonal question.
Yeah. So I will absolutely cop to it. You know, years ago, before we started really pushing this collaboration and communicating with each other, we would absolutely sort it and send it off because we just weren't thinking about it. You know, admittedly, it was just something that wasn't on our minds. And that would result in frustration at the GRC, and they'd come back and say, guys, this isn't tenable, you know, we can't do this. And so today, if I'm at the sorter and someone's sample looks very subpar, I will tell them, this doesn't look great, I'm gonna be honest with you. We're already here, I'm already running your sample, so I'm gonna sort, but I'm also gonna tell the GRC, and they can make the decision, right? If they don't like this, if they want to even go to the point of QC, then we'll do that. But yeah, so essentially I give the heads up at least at this point, because if I'm already sorting, I don't have any investment in not sorting for them. Maybe they can use the cells for a different application, take a pivot, probably not. But you know, if I'm there, I'm already there. My time is spoken for. And so I spend the rest of that time, though, talking with the investigator about, like, okay, so like magnetic depletion and like things you can do to clean this up, right? This is what you should be doing. Also, come to us and talk to us before you get to this point, because this doesn't work for you. And hopefully I nip that in the bud by kind of taking that moment to intervene. And I know the GRC folks as well provide their input to our investigators who have problematic samples that you know, they get to that point where they haven't really communicated with us, and it's just like all of a sudden we're sorting for genomics and we had no idea.
Could I add a tiny comment? Oh, sorry. Go ahead. So since my facility is so small and I'm so heavily involved, I've found that it's actually very beneficial that, like, when I'm involved with a project and I carry the weight of a PI, then I can tell people that I've said often that the most important service that we provide is to push the brakes on somebody's project. And when I tell, you know, the PI via text, actually that was a very good point as well, that direct lines of communication are important, because we joke all the time that it's like, what are these people doing, just airdropping it and then going to the moon? Like they need to comment, right? So a direct line of communication generally with the PI helps. And so I can text the PI and be like, you should not run these. And that's the most, and they like that, right? They like hearing it from me, and they're like, okay, we'll come back later. Generally speaking, you know, sometimes they're like, look, this is our only shot. These are only mice we're gonna get for a year. We'll do it. Okay, fine. But having the weight of the PI helps, or imbuing that weight upon an employee, where it, like, has to come from the leadership of the facility to be like, you know, what they say is the word of me, right? And so I have one employee like that, and that's really important. And then the other thing is, whenever possible, I drag that person into my
core facility and have them look at themselves in the flow trace or in the microscope, or we're doing spatial, something like that, and have them see it. Because if they make the decision like, oh boy, I don't like this, then like, oh, it's easy, right? So they got to see it too. That's the best, that they can see it. Yeah.
Yeah, I'll second everything that was just said, but I think importantly for us, it's that communication, right? So, you know, we're spread out between two cores, but we invest all that time up front with the investigator, through the consultation. We try to lay out, if we know they're going to be coming in with a sorted sample, it may not work perfectly. We try to be very clear if you're not going to hit 70% viability, if we're going to see dirty samples, if you're not getting out all your debris. These are the things we're looking for. And, you know, we don't have a lot of the physical space, so we kind of send people away, but we make sure on their submission sheet, we have a number to text and email that we can have real-time communication. We say, we're going to be talking to you in the next half an hour to an hour if things are not looking good and we need a go or no go. And that's where I'll usually step in if the folks actually processing the samples are thinking things don't look great. That's when I'll get on the phone and say, like, look, you know, I think we should pump the brakes. You don't want to spend, you know, $6,000 right now. You're not going to be happy with the data and you're going to come back not being happy. So yeah, I think it's really valuable, you know, point of view is like, that's our job, is to pump the brakes sometimes. Because I've definitely had investigators come and say, I sorted, I don't know why there's so much debris, and then we reach out for the flow plots, and it's like, yeah, they wanted to expand the flow plots, and okay, so I know enough then to say, look, because then sometimes it's just who's delivering the message, right? And if you can kind of tap in, you know, and kind of find common ground to say, yeah, I think we should probably go a different route, or we should probably modify. I think that is valuable, and we've had good successes with that. You know, some people, you know, they're just going to do what they're going to do, and you can't help that ever. And, you know, then we bludgeon forward. But, you know, most times people do want and respect the help we'll give them.
Yeah, thanks.
Don't plan any scuba diving trips 5 minutes after you drop off cells of the flow core.
Yeah, I've literally had people, like, I've caught them on their ride home. They've been like, oh, it's 4:30. We've already told them, please have the sample by 3, and they're on their driving home. And we're like, you fixed, you know, you put your samples in lysis buffer. I can't do anything for you, but I need you to know that. But yeah.
And also, if you need another opinion, if the samples are really crap, reach out to local field application scientists. They love looking at samples. Just, yeah, a little plug there. Yeah,
Thank you very much. It's very helpful and very applicable session for all of us. I'm running the Resource Center Flow Cytometry at Rockefeller University, which is very flat structure, very similar to Cold Spring Harbor in general, very diversified in terms of type of samples we are running, and we are practically not running immunology because they do have their own core, whatever they name, and that's another story. So my point is that sample prep, to be good, it's good suggestion. To have SOP, I wish, but we have training sessions to talk about general approaches, but when somebody is doing nuclei from ants' antennas, I can tell you it's quite a challenging project, which takes a lot of effort, not only from us with genomics. Data analysis seems to be the most straightforward for them, which is amazing because we are not part of this pipeline actively, which I wish we will, and we need to talk. But in general, having the sample, which has very low numbers of nuclei, let's say size of less than 1.5 micron, which people at genomics for 10x are using, you know, Countess. Okay, so let's not talk further. Very important to have genomics person understanding that cells matter, because very often genomics people think that DNA matters, but DNA is not coming, or RNA is not coming from bad sample prep and all of this. So that's another story. About the companies, full respect, but at the same moment, would you help with the sample prep of the ant antennas? I'm just in general saying that.
So
because when BD is jumping up and saying, come on, Rhapsody is your way to go, and when you start talking about challenging samples, that's another story. And I think where we have been put in by 10x in the point when genomics people start understanding single cells matter. It's very different, like, realm at this moment, and that's very important to understand, and your communication within Rochester. Thank you, Tim, for organizing everything. Is very important because asking researchers to put on CC both genomics and flow to be on the same side, it's important. But when genomics is saying, you brought me nothing, and nothing means, because on counters you see nothing, it doesn't mean anything. So what we are doing with our users, we saying we did buy the Luna fluorescent to be able to count cells, or nuclei in this case, saying that what you get, go. They go and say, please run, and at the end, in genomics, they get good data. And that's the only way, because genomics suggests spin down your sample to get a certain concentration, which is ridiculous, because if you have not many of the nuclei, what for to spin them down if you can download 42 microliters? You know, that's where I am, and it's very difficult, and I think your idea is amazing to have this very much streamlined. I think education is the most important between flow, genomics, and data analysis, which I think if Tim, with his power and everything, will be organizing some kind of very good, like, workshop. You know, not on Zoom, but in person, to have like brainstorming on the things will be amazing.
Actually, yeah.
Your support, Saima.
It's not support, it's pain. No, but
we feel your pain.
We all are interested in making things work. At the end, we want the projects work. And to convince the PI through the technician that things don't work. That's another thing, texting PI. I'm not texting my PIs. Because when somebody says, oh, come on, we have 26 Nobel laureates, it's another story, but that's not what I'm thinking of. Education for all of us, and cross-education through the steps of the sample prep and data analysis. That's important. And companies involvement is also important.
I think you're hitting on a lot of important points, and I think it's the communication, like we've talked about ad nauseum by now, but it's also the coordination, and it's educating our fellow cores, you know, on each of our subject matters and expertise. Like, we actually had a day where the whole Genomics Center went to the flow core, and they showed us some of the newer things so that folks who hadn't been active with flow could kind of see what we're talking about. We're talking about event counts and just the simple stuff we take advantage for those of us who've used flow before. We also coordinated about the time I joined the GRC, like having, when people sign up for a flow sort that's coming to the GRC, like a simple box that's checked so that it brings up a whole nother set of questions. It also flags for the flow core operators to know, like, okay, do an extra level of washing because of the sensitivity of the downstream assays. So like things like that we've laid the groundwork and then try to be very consistent with. But then when it comes to PIs, you know, again, it's always a push and pull. Some investigators are open to some mild critiquing and suggestions. Others are going to say, this is what we do it. I'm sorry, but that's what I'm going to give you. And I'm going to say, that's fine. But as long as you know, you give me a sample that's half dead, you're going to have limitations on the data analysis. And we try to do that, you know, cordially and nicely and educate. And then through other outreach, you know, we try to get out in front of that, and then leveraging what the companies tell us, right? Saying, you have to fit in these narrow set of parameters sometimes to make this work. We're happy to step outside that box when we need to, but here's what the potential issues are. And for the most part, that's fine. They don't always understand everything we're telling them at that point. You know, I'm talking about ambient RNA and I'm waving my arms around trying to describe what this impact will be on their data. Not as a bioinformaticist, but I've heard from the bioinformaticists. And usually that's like, yeah, we get it, okay, but I can't, but we can't
worst
we can't,
problem.
yeah, we can't do anything, right? So I think there's always a case-by-case issue here, but it does come down to, we're all at academic centers for the most part, right? So I think it's a matter of teaching, and it's a matter of kind of bringing them up to speed to where we need our users need to be, where our colleagues need to be, so we can kind of like do what we're asked to do.
I think there's a question behind you.
Thank you.
Hi, I'm Céline Lagesse from Cincinnati Children. I'm curious on the higher scale, like how your institution valued the effort you made in collaborating together, how they support you, did they recognize this collaboration? Is that used for recruitment of new faculty, like really at a higher...
If you want me to
Because I
answer
know,
this,
I mean,
can.
I'm a flow core manager, so I know we discuss with several of you to make it happen, but how the institution recognize that?
The institution doesn't recognize.
Yeah, I know.
That's the
But,
problem.
yeah.
Certain PIs are better than others, but this all comes from the team that is in CART and how we believe in making sure that the best science is being done. We're starting single-cell proteomics, so our proteomics team is talking to the genomics team to learn the lessons that they learn when they start at single-cell and hopefully won't make the same mistakes. They're going to find new mistakes to make. That all comes out of our institutional support, and they don't, I bury it in other costs so that we have the money to do these types of things. But, certain higher ups get it, but others don't, and same thing with PIs. I mean, there are PIs who want to buy their own machines, and it's like, no, because either it's going to sit there and no one's going to use it, or the data's going to come out crap because you don't know how to quality control it, or they're going to try to set up their own little core and get their buddies to help them so they can pay the service contract and get lots of crap. So yeah, it's an ongoing process.
What's your suggestion? What are you thinking about doing? Because they are lucky you are there to protect the cart, but how you can go to upper management?
I think the first step that the institution showed was letting us create a department. So we became a department and have the same kind of expectations and financials, and we continue to develop new ways to show people what we do. And it took us about 4 years, 3-4 years to find all the places where all the data was that we could then start to use to show the value of having these facilities. And when you only have about 35% of the papers citing the use of the facilities, we've got a little bit of ways to go. And I mean, we can have another ABRF all on that topic.
Yeah, thank you.
Can I just say one thing about that? One place you can get a little recognition for cross-core collaboration is if you're in a cancer center and in a cancer center support grant application, like I think it's looked upon somewhat fondly if you get the right reviewer. So it's like very big, but it can happen.
John, do you want to comment?
Yeah, it's actually following on that. So I think it's always a culture change, right? I think you take the winners where you have the winners, right? I think in our experience, we leverage what already has been built over the years to really bundle that into NCI, into that P30 grant. And our shared resources work really well because we can show the interconnectivity, and that was a real strength. And so when you have a win like that, you lean into it, and not everyone will come, but it's, if you get 60% of the community to be with you, I still think that's a win. And I think the other people that detract from that, let them go do what they're going to do. Just keep focus on building the cohesiveness that you can.
So you use the P30 call system to show that it could be applied?
Through that process, yeah.
Yeah, that's
That was a really challenging, but very fruitful thing.
I mean, Cold Spring Harbor, we have a NCI Cancer Center grant. They do encourage strongly that everyone who uses the shared resource to put this line in there. And it actually, I think the culture at Cold Spring Harbor is fairly good, and there's lots of acknowledgement and authorship. Like they're strongly encouraging authorship now for core directors who make a contribution. So I'm pretty happy with how we're doing it. But it has to come from the Cancer Center. Like, welcome to the Cancer Center fold, by the way. Yeah, but yeah, it has to come from the leadership.
Yeah, and I think building that community, right? We have now seminar series where we call it Omics and Donuts or Optics and Donuts for all the CART departments. Every few months we all identify investigators that we've worked with that have a publication recently or that we worked well with, who's a team player, and they'll highlight, we use flow, we use genomics, and we can see it. It's like, oh yeah, I remember that, and they're your champions, right? And then hopefully that trickles up, and their peers will see like, oh, you worked with the institute here and the resources we have to get that paper or that grant, and I think that helps, and we've seen it, I don't know, we've seen good feedback, I think, on it. And it's good to see from our end. We don't always get to see the end product, right? So it's always nice to see, oh, that's how that project turned out. So I found that really useful, and I think it helps to build a community.
I think we have one last question.
Great. This is Max from Howard Hughes Medical Institute. I have a general question for call management. I think especially for bioinformatics, we are seeing colleagues shifting to industry quite a lot. So is there any suggestions or how we could brand our core facility of to keep those talented people in place and keep them motivated? And,
I was asking you that question. No, it's really difficult, right? And what we found, unfortunately, is that a lot of the ways to keep really talented people is for them to have a passion for academic research and a reason to be in the location that they're in and not elsewhere, and that happens occasionally. But you have to raise a family and or whatever, if this is something you're interested in, you need to make that money, and sometimes academics isn't the way to go. So that's why we've resorted to what we have done, right, is to train the trainees how to do it, because we've just sort of resigned ourselves to the fact that staff bioinformaticians, maybe we can recruit a couple of people with faculty positions or something like that, but other than that, it's very, very difficult. I wish anyone else have any input on this in the room.
Well, I want to thank everybody for coming. Oh, I mean,
Go ahead.
yeah, go ahead.
Yeah, please.
I mean, this will give the time for the lunch lines to die down, so then you can.
Sorry. Okay, so I'm standing between everybody and lunch. That's bad. So, so I'm Brian from Arc Institute. So on that point, I was wondering if do you just resolve to say, okay, so we are an institute where we're just going to hire maybe bioinformatics people who are a little bit more junior, train them for 3 years, and they're going to leave, and then we plan for that. So have a rotation, so that there's no knowledge gap. Does
That's effectively how we're running the bioinformatics shared resource here, and how my core, I have bioinformatician people in my core, right? And so that's how we try to do it, is to have overlap.
Yeah. But the other thing, so the other thing I think, so at ARC, what we're trying to do is we really push the RA. So these are wet lab RAs who might also want to go to industry and so on. But we really push the RAs to the project of the PI groups really early. So basically when they are planning their project, we say go join their group meetings. Are you interested in go really participate? And then when they're wet into the biology, and then we'll tell them go ask a PhD student to give you their slide deck, or sit down and explain to you what they're doing, and that kind of thing usually. Yeah,
we do that too. You do a long-term sort of embedded collaboration.
Right.
That's critical. You stole one of our bioinformaticians, by the way.
Oh, Alex?
Again, thank you all for coming. There is the DEIB networking lunch, as well as lunch in the session, and look forward to seeing you the rest of the conference.
Thank you.
Yes.
I'm going to defend the owner of your information base.
Oh boy.
I've had, you got somebody that has been dangerous.
Yes.
Sure.
You use 2 options. The idea's been floated.
Yes.
Yeah.
I'm probably going to be the prototype for that.
Lucky to have.
So we're working up to it. Exact timeline, I don't know, but I think that's our next step to kind of build that support even more.
We have enough touch points that it makes sense. We've started the conversation. Steve's the identified person, because you need to, because not everyone has this